I think we are seeing a very interesting phenomenon where, we were mainly focused in the treatment of patients with high-risk disease, but now we're moving actually into quite a bit of studies for lower-risk disease and actually some studies for transfusion-independent low-risk MDS and also clonal cytopenia of unknown significance. So I'll go quickly through some of the studies that we have at MD Anderson that I will be discussing at the meeting. So we have studies with canakinumab. This is the interleukin-1 inhibitor for CCUS and low-risk myelodysplastic syndrome. We're working on IRAK4 inhibitors, in particular the Rigel compound, also for lower-risk MDS. We're trying to target inflammasome with NLP3 inhibitors, such as the DFB compound. This is an asset from Novartis. As I mentioned earlier on the first question, we have now studies going phase 2 with the oral azacitidine cedazuridine. This is trying to model PK/PD-wise compared to IV subcutaneous. Moving to high-risk disease, we have what we call total oral combinations with, for instance, oral decitabine with venetoclax. This had already been published. They are very effective. We're moving into combinations of chemotherapy, for instance, low-dose cladribine, cytarabine, venetoclax for high-risk disease. We have a second IRAK4 inhibitor. This is the Curis drug that is for high-risk MDS. And then we have some targeting approaches, for instance, with quizartinib in combination with an HMA for FLT3 mutated or CBOL mutated MDS. We are now starting a study with SL401. This is a CD123 approach that is targeting actually patients with P53 mutated. And there's now interest on developing drugs that block something called CLIC, that is a key enzyme regulating splicing. So we're talking about the Cordia compound that is also starting in patients with advanced myelodysplastic syndrome. We are kind of deploying lower doses of CPX. This is going back to the cladribine, cytarabine, venetoclax approaches for high-risk disease, now in combination also with venetoclax. And to finalize, we're actually moving into two studies that I think quite interesting with luspatercept. One is an IST here at MD Anderson looking at patients with very early stage disease. And there's also an international trial known as Elements that is quite exciting for transfusion-independent low-risk MDS, comparing luspatercept with ESA. And then finally, we are ready to open studies looking at NRAS-mutated disease with ERK inhibitors. And we have a couple of studies for IDH1 mutated disease. Now, this sounds like a lot, and it is, but we are lacking studies for P53 mutated disease, and we're really lacking studies for second-line HMA failure disease. So, all these things are in transit, but we need to have some clear kind of light after we finish all these things to really go for some phase 3 type of approaches.